टेम्परेचर थोड़ा बढ़ा दे
16 ज्यादा ठंडा है तो
25 का तो ना 25 का कंफर्टेबल ये तो कम होता होगा 22 होता होगा इमोशन okay so yesterday also we had a class what is that what we can start that way
drawing of the heart.
drawing of the excretion we started excretory system we started structure of the nephron structure of the kidneys we are to study Pick out your pencils. We first have to start with this. Now the excretory system of human. We are going to start with this. The same chapter. You are absent, no? 2 days you are absent. 3 lectures you are absent.
actually starting with one new chapter.
A new chapter meaning?
How to work to produce that chapter name there.
So how does it matter?
I'm पूछे
मतलब तुम नया चैप्टर होते तो आते ऐसा है क्या?
I was asking that
ये question you are not supposed to ask. This is what we are going to decide. No, as and when the chapters get, you are supposed to check whether I have a lecture today. I have to go for the lecture. Finished.
I thought the new chapter started.
No. We are studying the process of excretion. Yes. Let us start drawing the excretory system in human.
And we were also about to draw the nephron.
हाँ पहले ये करेंगे उसके बाद नेफ्रॉन करेंगे You all had lectures from 7:30, no?
Yes, ma'am.
7:30 सब आए थे
You said
और हाँ all of you were present 7:30 lecture you were there why did you remain absent for the last 2-3 days 2-3 lectures okay so take out your pencils the way how to draw diagrams remember only with a pencil to draw when you draw the kidneys, one specific thing to remember is both the kidneys will not draw at the same level. The right kidney is supposed to be a little bit lower. So take a look. When you draw the diagram, the right kidney has to be a little bit lower. Start your diagrams, all of you.
I'm not sure.
Who is talking? This one is constant.
Yes, yeah.
Not that small. I repeatedly told you, erase, try again. Why so small? It's too small. Too small. It should be at least this way. I always told you diagram half of the page में draw करना है. Should not be that small.
Yes, sir. These are clear.
After you draw this diagram, you will leave the rest of the page, and then the next page you will draw that diagram. Next page you will draw that.
What's that? Muscle.
Sphincter. SPHINCTER. When you start this diagram, this set of blood capillaries, just draw with pencil the black parts that I draw. Made it very clear that you have to draw this part little bit bold, dark draw karna. And this red blood vessels that I draw, you can draw a little bit light. Don't use colors.
What? That's good.
Open the order. Okay, fast, fast, fast. Both the diagrams you will draw in two different pages, okay? Not in the same.
Yes. Under that.
Hmm. No, that will not open. It's a problem. You have copied it.
That could be employed.
This.
That's it.
ये जो numerator you will take now, you will not take straight from here. You should take like this.
ये होगा बड़ा देना है
By the other red pencils. The other red pencil आता है ना? The pencil body and the thick needle is not red.
मैम इतना ही होगा
No, I don't want this actually. I want a structure of a nephron. That is what I want to show. This diagram is simple. अभी ये जो दूसरा you will draw here, but it should come here. Nephron. It should come here. Now you can say.
Oh my God, चाचा इधर ना
Blood vessels and discharge is okay.
ये
Kidney का shape.
ये
Kidney का shape. The shape of the kidney always should be means Rajma means कैसे रहता है? Concave. And this side should be convex and concave. हाँ.
हाँ तो सिखाते
Shape
हैं
of the kidney should always be this.
and it has because के
Because all curves.
सब आता है
Shape of the kidney is not straight. Blood vessels are okay. Ureters that you draw. ये ठीक है. Yes, it's straight. It's not. That's actually what is straight. थोड़ा सा modify कर लो. Blood vessels are straight करो. Shape of the kidney थोड़ा सा proper. थोड़ा proper diagram better कर.
इधर क्या ही बनोगे
Pencil. The shape of the kidney that you draw should be always this way. इस तरह का shape होगा. Flat. Okay. Then blood vessel, ureter that you draw इस तरह से आएगा. Don't draw the ureter straight like this. Okay. And blood vessel. यहाँ पे क्या है? The shape of the kidney is little bit proper कर लो. Blood vessel थोड़ा सा लगभग ठीक है. Shape of the kidney थोड़ा सा proper करो. इसका ये करो fast. This is the diagram that you are drawing. This is the diagram there in your textbook, but the diagram of your textbook is not very clear. If you check, the parts are not understood at all. Complete the diagram first. The first diagram you complete, then I will explain this parts. Then we will go to the next one. Those of you are done with the first diagram, give it to me. All of you, are you all done with the first diagram? First diagram is over. Yes. Complete the first diagram. We'll go to the second after some time. Let us learn about the first. I had started the concept the other day already. This is the diagram of the excretory system. The excretory system in human. So now this excretory system, all of you look into this diagram. All of you look here. All of you. Toshita, no? No talking. This diagram drawing hardly takes 5 to 10 minutes. We have taken too long. Now, when we are talking about this, what are the parts names? I already mentioned some the other day. Let us again do it. This is the excretory system of the human. What was the location I told you? What is the location? Present above the hips. The two kidneys that we are mentioning, the two kidneys are present where? The two kidneys are present on either side of the vertebral column. Location of the two kidneys are on either side of the vertebral column. This entire system, majorly that I talk about the kidneys are present where? They are present above the hip. But this bladder is actually present at the lower abdomen, exactly where the intestines end up, exactly at the lower abdomen. So, what are they made up of? This entire kidneys that we are talking about made up of muscles and majorly smooth muscles and some connective tissue fibers. Majorly smooth muscles, connective tissue fibers, the entire structure. Next is, when we are now looking into this excretory system, the other day already we mentioned that talking about the function of the kidneys, two functions of the kidneys if I have to mention. The first function of the kidney that we had mentioned was filtration of blood. So now, looking into this aspect, we have to talk about that this kidney should be getting adequate blood supply. So, but which are those blood vessels? Now see the label, all the labels are here. Which is the blood vessel which is carrying blood towards the kidney? The renal artery. Which is the renal artery? This is the renal artery. The renal artery is carrying the blood towards the kidney. So the blood which is carried by the renal artery is oxygenated. We are talking about an artery. The blood carried by the renal artery is oxygenated, but at the same time, it has got rich concentration of nitrogenous waste substances. So which are the nitrogenous waste substances? Now that I talk about filtration of the blood, filtration of what actually we are to think about? Filtration of urea and uric acid we are majorly thinking of. Filtration of what we are to understand? Filtration of urea and uric acid. Two major nitrogenous waste substances. They are very, very toxic in the body, and if they are present in the body for a long period of time, they start affecting other tissues and organs. So first, talking about the kidneys, they get blood supply from the renal artery, which is a branch of the aorta. That's why we have mentioned the aorta and the vena cava also. Now talking about the veins, if I have to mention, what is this blue vessel coming out from the kidney known as renal vein? And like any other vein from different organs of the body, if this is a vein which is coming out from the kidneys, like the veins coming out from the stomach and the liver and etc., all other veins of the body, like the one that the kidney also gives away, that is the renal vein, they will always unite to form the big broad veins of the body known as the vena cava. So that is about the blood supply, always very clear. What are the rest of the parts? The rest of the parts, a pair of kidneys, just now I said. From each kidney, a pair of narrow, long tubular structures emerges out. And this long, narrow tubular structure is broad when it starts from the kidney. So these are additional parts for you, not necessary for labeling. Later when we understand that process, little bit one of those terms will be necessary. So I'm just mentioning them. Now in your label, no need. What is this broad end of the ureter known as? The broad end of the ureter is also known as the renal pelvis. So I would say that the ureter starts from the kidney as a broad tube known as the pelvis, and then as it emerges out, leaves the kidney, it narrows down. It narrows down, goes down towards the lower abdominal region to enter into a muscular sac-like structure. Muscular, what type of muscles again this urinary bladder is made up of? Again, it is made up of smooth muscles. So this urinary bladder is made up of smooth muscles. So now that I say urinary bladder is made up of smooth muscles, the first thing that comes to your mind is, yes, it is involuntary. It is an involuntary structure. So this urinary bladder is made up of smooth muscles, and it will be collecting the liquid waste by the way of the ureter from the kidneys. And then the bladder continues as a short tube known as the urethra, also made up of smooth muscles. And on the inner wall of the urethra, or say at the junction of the bladder and the urethra, there are some muscles. More or less, these are also acting like the valves that we have to mention. Now, what are the job of these pair of muscles? What are these muscles doing? These muscles will be relaxing and contracting from time to time. Relaxing when? Relaxing and closing down. They will be relaxing and closing down when the bladder is empty or not absolutely filled. When the bladder is absolutely filled, or when the bladder is filled to a great extent, these muscles will contract and they will become pulled and constricted towards the wall, and the passage has been formed. The passage has been formed for emptying the bladder, a process known as urination. So now that we are talking about that, what is the job of the urethra? To release the urine out from the body. And I'm saying filtration of the blood is the most important function of the kidney. Filtration to give rise to what? Filtration to give rise to fluid, the excretory fluid of our body, the excretory fluid of our body known as the urine, which is rich in urea, uric acid, having a very high concentration of urea majorly. Next is, after you are more or less aware about the blood vessels, the renal artery, renal vein, all these parts, we were also talking about one more concept, that is, look into the kidney in a section. Then what do you come across? If this kidney can be cut in a longitudinal section, then what do you come across? We come across that the kidney has two distinct regions. And what are these two distinct regions known as? The two distinct regions, outer region which is darker, known as the cortex. Inner region which is lighter is known as the medulla. So what are cortex and medulla in a kidney? If the kidney is cut longitudinally, the entire kidney shows two distinct regions. The outer region is darker, the inner region is lighter. Now the inner region, if you have to draw, there are quite a number of structures in the inner region. There are quite a number of structures, and then all these tubes, and then so now the inner structure, when we are talking about, there are a huge collection of some tubules. Talking about one such tubule, as yesterday also I mentioned, looking into these granular structures. What are these granular structures? If I want to draw just one of them, bit prominent, the structure would be something like this, where each of this structure is roughly like this. Where I want to see that what are these bead-like structures and what is the relation of these bead-like structures with some fine thread-like structures all throughout in the medulla, we come across that actually these bead-like structures are something like a head-like structure that is present in the cortex, followed by some thread-like structures long down in the medulla, and then again come up. So what is this entire structure now known as? We want to know what is the name of this particular structure, and they are present in millions in the kidney. And this type of structures, tubular structures, present in millions in the kidney. What are these tubular structures known as? Nephrons. We had discussed this the other day also. What are nephrons? Nephrons are the structural and the functional unit of the kidneys. Why is it mentioned as such? Structural unit, why? Because they are of the same structure. Those 2 million nephrons in both the kidneys that we are talking about, same structure. And why functional unit? Because all of them are performing the same job, filtration of the blood. Now, what are the parts of the nephron? What are the parts of the nephron? This was the structure that here we had drawn simple. Now, magnify, elaborate to draw the structures. What are the parts? This is that head-like structure that we are talking about, which is present in the cortex of the kidney. And this is the entire tubular structure that we are talking about, and where a whole lot of processes takes place, talking about the the filtration of the blood to form the urine. Now we have to learn what are the names of these parts and how does the process of filtration of the blood take place. So there are also a few processes you'll have to come across, few terms you'll have to come across. So let us start. Labeling the parts, first of all, the names are very important. Now if I start talking about the parts of the nephron. The nephron starts from the cortex, goes down till the medulla, and again to the cortex. So I would say the nephrons, millions of thread-like structures are spread out in the entire kidney. Roughly, I told you the number, every single kidney, one single kidney will have millions of them. Now let us label the parts of this. This is where we start with. Now this particular vessel which is carrying blood towards the kidney. What did we name this vessel as? This is the renal artery. From there we are starting. This labels, what is the name of this vessel? This vessel is the renal artery. This is the renal artery. Now already you are aware, the renal artery carries blood towards the kidney, towards the nephron. Towards the kidney, more specifically towards every single nephron. So the renal artery carries blood towards the nephron. But as the renal artery moves towards the nephron, the renal artery again divides into branches. Can you see? The renal artery is divided again into branches. What is the branch of the renal artery known as? The branch of the renal artery is known as the afferent arteriole. These names are new for you. You'll have to remember these names. What is this branch of the renal artery known as? The branch of the renal artery is known as the afferent arteriole. Check the spellings, write correctly. Afferent arteriole. This afferent arteriole now divides into a network of blood vessels. Can you see? This afferent arteriole, the moment it enters into the nephron, starts dividing into a network of blood capillaries. What is this network of blood capillaries known as? This is known as the glomerulus. This is known as the glomerulus. How do we define glomerulus? What is glomerulus? A network of blood capillaries formed from the renal, from the afferent arteriole specifically to write that always. What is glomerulus? A network of blood vessels formed from the division of the afferent artery. What next? The Bowman's capsule is actually present or lodged. It is said that the glomerulus is lodged. Lodged means what? It is fit inside. The entire ball of blood capillaries, where is it fit inside? If you see, it is fit inside a cup shape. This is the diagram I am showing you because this is what is almost close in your book that is given there. This is a diagram which is there in your textbook almost. Now, when we are talking about this double-walled cup-shaped structure, this double-walled cup-shaped structure is a structure where the glomerulus is fitted. So, what is this double-walled cup-shaped structure known as? This is known as the Bowman's capsule. The glomerulus and the Bowman's capsule together is also known as the Malpighian body. Is known as the Malpighian body, or there are other names also, renal corpuscle, etc., etc. All of you, as you draw the diagrams, now understand and start labeling the parts also. So we are talking about the glomerulus, a network of blood capillaries which is fit inside a double-walled cup-shaped structure. What is this double-walled cup-shaped structure? Bowman's capsule. Glomerulus and Bowman's capsule is always together, named as the Malpighian corpuscle or the Malpighian body. Come down. Next is we have to talk about the tubular part. But before we talk about the tubular part, we'll finish with this blood capillaries. The blood capillaries are formed by the division of the renal artery. But on the other side, this blood capillaries will again unite and come out of the nephron. So what is this blood capillary known as? And can you find out the difference between this blood capillary and this blood capillary? Are you able to find a difference? Always check whether you have drawn also that way. This blood capillary always you'll have to draw little bit broader and this is thinner. Okay. When you draw the diagram also take a note. This has to be broad and this has to be narrower. What is the name of this blood vessel? The name of this blood vessel is efferent arteriole. This is known as efferent arteriole. The first arteriole that we had written was what? We had written down the afferent. And this is efferent. Take a note. This is efferent. And here we had written afferent. Capital A and E. So what are we naming? The afferent blood vessel, that is the afferent arteriole and efferent arteriole. And what are these blood vessels? These blood vessels that we are talking about, these are known as the secondary blood capillaries. Secondary blood capillaries. Now we label the entire tubular part. Now this entire tubular part as such, if I talk about in your book, they have only mentioned as the tubular part. There is no differentiation of the names. But I will explain you with the names because without the names, when the entire story I talk about as tubular part, you will also get confused which tubular part man is talking about. So we generally don't explain that way. You are supposed to remember the names of the three parts of the tubule. So it's very easy to remember also processes. So what are the names of these three parts of the tubule? The three parts of the tubule. The first is, I understand this part of the tubule, this part of the tubule. This part of the tubule. What is this? This part of the tubule is known as the proximal. This part of the tubule known as the proximal. Proximal means first, the first region. Proximal convoluted tubule. Proximal convoluted tubule. Next is this long, narrow, U-shaped tube, which plays a very, very important role when we are going to learn the process. What is this tubular region known as? This is known as the loop of Hanley. This is known as the loop of Hanley. What about this particular region? The next curved coiled region. This next curved coiled region that we'll have to remember. This is known as the distal convoluted tubule. Convoluted, get a try understanding the meaning of this word, distal convoluted tubule. What is the meaning of the word convoluted? Extensively coiled. Convoluted means coiled, extensively coiled. Go and wash your face. Extensively coiled. So now we are talking about the first one. Are you clear about the renal artery and the afferent arteriole? What is the relation, tell me, between the renal artery, afferent arteriole and glomerulus? What is the relation? Renal artery divides to form renal artery divides to form afferent. Renal artery divides to form afferent. And afferent further divides to form glomerulus. And the capillaries of the glomerulus unite to form efferent. And this again divide to form capillaries. Clear about the blood vessels? And this capillaries will again unite back to form the renal vein. Done? So the blood vessels, you are done. Now let us talk about the tubules, the parts. Glomerulus, network of blood capillaries formed from the afferent arteriole. What was Bowman's capsule? I told you it is a double-walled cup-shaped structure. A double-walled cup-shaped structure. So this double-walled cup-shaped structure, we will learn features. The double-walled cup-shaped structure. Say suppose I take a thin ball. I take a thin ball. यहाँ पे इस ball में यहाँ पे I punch it. You punch this ball and try to make a depression. क्या होगा? The ball will take a shape something like this. The ball will take a shape something like this. Now that the ball has taken a shape something like this, what is happening? The inner region has become, it is becoming pocket-like, no? A depression ban gaya. So this inner layer, now we have made it porous. We made it porous. If that is a structure that we are thinking of, outer lining to same smooth without any pore tha, and now we have started making it, punching it karke, and then we started developing a depression there. So is there a space, hollow cavity between the inner, between these two layers? Cavity hai ki it is filled with any solid substance? Cavity hai na? So same like that. What I'm telling you right now is, I'm telling you, think of anything, how to understand what it, because the entire process is based on the texture of the cup-shaped structure. I say, if at all we are thinking of a ball, a soft ball, and then we punch it inwards, what are we going to get? Yes, a depression may be formed. And now that a depression is formed, what are we doing is, we try, now that a depression ho gaya, now that we try making some small holes in it. We try making some holes in it. So ये जो inner line and outer line है, between the inner and the outer line, there is enough empty space. It's just filled with air, nothing else. Same is the structure when we talk about the Bowman's capsule that we have inside our kidneys. एक inner line है which is porous, एक outer line है which is solid and thick, and between the inner and the outer line कुछ भी नहीं है. It is just empty. Okay, it is just empty, but difference abhi yahi hai that this outer line will be followed into a tube-like structure. That is the tube that we are talking about. So what are these three parts of the tube now? The three parts of the tube, the first part of the tube is called proximal convoluted tubule. You understand what is the meaning of tubule? Tubule matlab narrow tubes. Convoluted matlab, what is the meaning of the word convoluted? Coiled, extensively coiled. Distal means far away. Far away किससे far away? From the Malpighian body. Malpighian body से जो close tubule है, जो पास वाला tubule है, the tubule which is near to the Malpighian body, उसको बोलेंगे proximal. Proximity का मतलब पता है? Closer by. So proximity, proximal always means close by जो है. So what is the meaning? Proximal convoluted tubule Q. What is the name? Why the name? क्योंकि this tubule is coiled and this tubule is close to the Malpighian body. Distant का मतलब क्या है? Distant, far away. So this distal मतलब from the word distant. Distal मतलब far away from the far away from the Malpighian body, but this is also convoluted. Convoluted का मतलब है coiled, just like the small intestine, extensively coiled and wrapped and coiled and wrapped. इस तरह के tubule है. So now this distal convoluted will end up into a fine, long, bristle-like structure, which is also a long tubule. What is the name of this tubule? The name of this tubule is the collecting duct. The name of this tubule is the collecting duct. So once you are aware about the terms, the position, the relation of all these terms, then only the process will be able to understand. Nothing very difficult, the three major steps to come across. So now I talk about the renal artery. Renal artery, when it goes near the nephron, the renal artery divides into a finer tube. What is the name of this? Afferent arteriole. This afferent arteriole will divide into a number of blood capillaries. What are these number of blood capillaries? Known as glomerulus. Where is the glomerulus packed? In a double-walled cup-shaped structure made up of smooth muscles, Bowman's capsule. Completing the blood capillaries first, these blood capillaries will again unite back, resulting in efferent, then a network of blood capillaries, and then again uniting back, forming the renal vein. Let us just talk about a difference between renal artery and renal vein here. It is a vein, so definitely what type of blood should it carry away from the kidney? Deoxygenated. And this is a renal artery. Very clear, try understanding, yes. The rules are same like what you had studied for the heart. If it is an artery, it carries oxygenated blood. If it is a vein, it carries out deoxygenated blood. Yeah. With the kidneys, what type of purification are we talking about? With the kidneys, we are thinking of that this blood, though it is oxygenated, rich in oxygen, but it has got high amounts of urea also. That is what is getting filtered here. So the blood which is coming out from the renal vein, should it contain less urea? Always. The blood which is in the renal artery going towards the nephron should always have or always has more of urea, more of uric acid. Though it is oxygenated, purification is nothing to do with oxygen here in the kidney. Purification is supposed to be done with the urea, uric acid and many other poisonous chemical substances. And then the tubular part, the proximal tubule, between the proximal and distal, name the long U-shaped tube, loop of Henle. And then a very important process taking place here, we will learn this. And then the distal convoluted tubule going and joining with the collecting duct. Now from the collecting duct, if I have to draw a connection, the collecting duct, if I have to draw a connection with the collecting duct, I would say what parts now? After the collecting duct, what is the relation of the next parts of the excretory system? The collecting duct will enter into the pelvis, or the collecting duct will join the pelvis. Now you know which part is the pelvis. Which part is the pelvis? This part is the pelvis. So every collecting duct will join the pelvis. Then pelvis will join what? Ureter. And ureter will join the urinary bladder. And the bladder will join what? The urethra. Are you able to understand this?
Yes.
So what is the connection of that diagram and this diagram? If I talk about where is the last part of the nephron? Collecting duct. तो कलेक्टिंग डक्ट के साथ ये डायग्राम का क्या रिलेशन है? कलेक्टिंग डक्ट will empty the contents of collecting duct. Where is the collecting duct? Again, if I draw this is the nephron and then coming up. So यहाँ पे है collecting duct. So this collecting duct will throw the contents in the pelvis. Pelvis से the content is by the ureter will come down into the bladder and released out by the way of the urethra. So this is the relation of the collecting duct which is there here and the rest. यहाँ तक आया समझ में?
Yes.
Any doubt so far? The system is there, parts, location. So once you are aware about this, and inside the nephron, inside the kidney, the nephron, that. Now, the process. If you have to understand the process, what are the three main steps involved in the formation of the urine? But formation of the urine ke liye, we'll have to majorly think of what? For the formation of the urine, we have to majorly think of the entire nephron. Filtration of blood is happening and formation of the urine is happening. So to understand the process of formation of urine, we are talking about process of urine formation and what are the steps. Process of urine formation, what are the steps? The three most important phases are, the first phase is known as, the first step is known as ultrafiltration. The second step is known as selective reabsorption. The second step is known as selective reabsorption. The third step is known as tubular secretion. So these are the three steps that we are going to learn now. Ultrafiltration क्या है? Selective reabsorption क्या है? And tubular secretion kya hai? Where are these processes happening? All happening in the nephrons. Every single nephron, millions of nephrons that we have, going through all these three processes simultaneously, and that's how the blood is getting filtered from time to time. Let us start understanding the first step, that is ultrafiltration. To understand the process, again I am drawing the same structure in a simplified manner, in a elaborate form. The entire structure again I draw. I start with the Bowman's capsule. Let us start with the concept. You will not draw this diagram. So I am talking about, this is the Bowman's capsule. The Bowman's capsule and the three parts of the tubule. So we want to understand the process. So to understand the process, let us come across the Bowman's capsule, and there should be the blood vessel. So we are talking about the blood vessel. Blood has to pass through this. So let us think of a blood vessel here. Which blood vessel? The renal artery, the glomerular capillaries, and the renal vein. But majorly we are now talking about the afferent coming in, going out by the efferent. So let us talk about this. The afferent is broader, and the glomerular capillaries, and the efferent is narrower. Okay. So afferent arteriole we had in the diagram I mentioned. When you draw the diagram, this has to be clearly mentioned. The afferent arteriole, which is this arteriole I am drawing the afferent arteriole. Which is this arteriole I am drawing the efferent arteriole. Which is this tube I am drawing? Which is this blood vessel I am drawing? This is the glomerular capillaries. Now I just think of one such capillary. I'm just thinking of one such capillary now. Try understanding. So now that I talk about this blood vessel, now this is the afferent arteriole. As it enters, the afferent arteriole carries blood. The blood is flowing in this afferent arteriole at a great pressure. The blood that is flowing in the afferent arteriole is at a great pressure. Does the blood flow in an artery at a great pressure? Does the blood in an artery flow at a very high pressure?
Artery is when a high pressure.
Does the blood flow in an artery at a high pressure?
Yes, ma'am.
What are the reasons? We have studied that. What are the reasons? Because of contraction of the left ventricle of the heart. What is the second reason? Blood flows in an artery at a very, very high pressure. What is the second reason? The section of the artery, do you remember I showed you?
It is.
The inner space is very narrow. The lumen we mentioned. The second reason is why the pressure of the blood in an artery is so high, because the lumen is narrow. What was the third reason why pressure of the blood in an artery is very high? Valves तो नहीं है, valves absent. So valves are absent, and what was the third reason to mention that pressure of the blood in an artery is very high? Most of the arteries carry blood downwards. Downwards. So gravitational force भी है. So all these points you'll have to remember. So right now I think of that this is the afferent arteriole. This is the afferent arteriole. Which blood vessel am I thinking of? This is the afferent arteriole. Remember, carrying blood from the renal artery, the blood flowing in the glomerular capillaries. I think of one such capillary now. Are you able to understand? Are you able to see this tubule? Let me draw a bit bigger. I draw a bit broad. Try to understand. I'm thinking of only one such capillary now. So what are some of the features of the capillaries? Earlier also we had studied the blood which flows in the capillaries. If the blood in the artery is very high pressure, the blood in the capillary is also very high pressure. So now that the blood which is flowing in the arteries, in the capillaries at a very high pressure, what should happen to these capillaries? The liquid part of the blood oozes out. Okay. So try understanding, try following this stepwise. What am I mentioning about the concept of ultrafiltration? Now that the blood is flowing at a very high pressure, the blood is running and forcing these capillaries. As it is running through these capillaries, since the capillary wall is very thin and the capillary wall has got small pores in it, some amount of the blood, meaning amount of the blood means which component of the blood is oozing out. Some liquid part of the blood is oozing out. And which is this liquid part we are now thinking of? The liquid part which is oozing out is majorly the plasma. So which is this liquid part of the blood oozing out? Which is this liquid component we are talking about? It is the plasma which has a whole lot of plasma with the proteins. Plasma with a whole lot of proteins. Plasma with a whole lot of salts. RBCs, are they coming out ever? No, they will not. Some few WBCs can come out. That was a concept of limb that we had studied earlier also. Here also the same, but right now we are not bothered about the WBCs here. We are only bothered about all the components of the plasma. I repeat what I mentioned. The blood is running in the capillaries at a very high pressure. The blood brought by the efferent arteriole. Very high pressure because it is coming from the renal artery. And the blood running in the capillary of the glomerulus, which is this capillary I am showing you. I am showing you one such capillary of the glomerulus. Glomerulus is a network of hundreds of capillaries. I am just showing one of them. What is happening in one of the capillary? Blood as it rushes out, what is happening? Some liquid component starts oozing out. What is that liquid component? Plasma. What does the plasma contain? Whole lot of minerals and proteins and salts and ions, etc. But what will happen if you remember this particular structure I was mentioning, and I said that if I now punch here and the depression made, and if I now put water, what will happen? अगर यहाँ पे पंच करने के बाद जो इनर मेम्ब्रेन हुआ, जो ऑलरेडी दब गया है, प्रेस्ड हो चुका है, डिप्रेस्ड हो चुका है, now we make some holes and अभी उस कॉन्केविटी में, उस कैविटी में now we put water, क्या होगा? सीप इन, वाटर अभी सीप हो जाएगा, सीप होके यहाँ पे कलेक्ट हो जाएगा. एंड अब एक्सटेंशन ऑफ दिस बॉल आई एम सेइंग दैट हमारा किडनी लगभग इस तरह का है, where the broad end of the ball has got a tubule like structure also continuous, so definitely it will keep on flowing. यही होना चाहिए, यही होता है when I talk about a nephron. So now that in this glomerular capillary, since the blood is rushing at a high pressure, certain liquid component ooze out हो गया. Ooze out होने के बाद वे are they collecting? They are collecting in the cup. ये जो depressed cup है of the Bowman's capsule, वहाँ पे they are all collecting. But the inner region is all hollow and it is continuous. So now they are all flowing. They are all flowing. All these fluid, they start flowing in this hollow cup-shaped structure, and then continue into the hollow tubule. ये जो पूरा tubule है, it is hollow from inside. Same like the intestine, hollow from inside. So now, what is this fluid known as? What is the name of this fluid? If I say that this is the fluid which is getting collected in the Bowman's capsule and उसका composition लगभग ये है. What is the name of this fluid? The name of this fluid is glomerular fluid. The name of this fluid is glomerular filtrate. And what is the name of this process? Ultrafiltration. Q ultrafiltration? Because the filtration of the blood is happening because of the high pressure of the blood. You think of a water pipe again. At one end of the water pipe tap के साथ connected है, and the water pipe you draw it somewhere. And water pipe में बहुत सारे holes किया हुआ है, and you turn on the tap high full force. What will happen? The water will just sprinkle, no sprinkle. वो holes में से sprinkle होगा. लगभग उसी तरह का एक process हमारे nephrons में हो रहा है. So the blood is rushing in these capillaries. A huge amount of the fluid starts oozing out. अब ये जो fluid ooze out हो चुका है, where is that fluid now? The fluid is all collected in this cavity. ये जो cavity है, ये जो एक depressed region है of the Bowman's capsule, यहाँ पे the entire fluid is there. But ये जो cavity है, it is not solid, it is porous. I have mentioned always, see there in the diagram also we draw, this entire inner wall is porous. So जो भी fluid यहाँ से निकलेगा, first of all it will collect and then start seeping down. Always it will start seeping down. यहाँ तक आया समझ में? So now, what is the concept of ultrafiltration? So I say, to understand the concept of ultrafiltration, what are some properties of the Bowman's capsule we understand? We understand number one, the Bowman's capsule is porous like this. The Bowman's capsule is not a continuous solid thick layer. Second is what I said, that if these are the blood capillaries, and then I understand that the blood is rushing in this capillaries at a great force. But ये सारे जो capillaries है, these capillaries में से on the wall small pores are always, that is always a feature of the capillaries. Capillary wall is always porous, whichever part of the body, खाली kidney में नहीं, everywhere. So now that the blood is rushing here, definitely some liquid component starts oozing out. So this liquid component which is oozing out, what is this liquid component known as? Or what is this liquid component actually? It is the plasma. Plasma with whole lot of proteins and salts and minerals, etc. But what will happen to that liquid now? That liquid will first collect in the inner hollow and then it will start moving inwards. Then it will start moving inwards and start moving into the hollow of the tube. What is the name of this process? The name of this process is ultrafiltration. Where is ultrafiltration happening? Ultrafiltration is happening in the glomerular capillaries. What does ultrafiltration give rise to? Glomerular filtrate. What is the composition of the glomerular filtrate? Plasma and some proteins and some salts. But why does the blood cells are not able to come out? Why are the blood cells not able to come out? The blood cells are not able to come out because the pore size does not allow the RBCs to come out. The RBCs would get stuck. The RBCs does not come out. So now that the blood rushes out, now think, the blood which is coming by the way of the afferent arteriole has given away a whole lot of water. Isme plasma, I had written plasma with protein, salts and एक particular component जो maximum quantity में रहता है, that is the water. How much of this glomerular filtrate is formed per day? Per day, almost about 160 to 180 liters of glomerular filtrate is formed. Per day, if I think of all those millions of nephrons, how much of this glomerular filtrate is formed? Almost 180 liters, because the blood, every small amount of blood goes to the nephron almost 300 times a day. 24 hours में एक specific amount of blood, if we can keep a trace, एक छोटा सा specific amount of blood, maybe 1 ml, 2 ml we are tracing, that will go through the nephron almost 300 to 400 times, and that is how we understand एक 180 liter of glomerular filtrate ही बन रहा है, but we understand blood in our body is just 6 liters, so 180 liter filtrate कैसे? Because एक ही specific amount of blood दिन में खाली एक बार प्यूरिफाई नहीं हो रहा है, almost 300 times प्यूरिफाई हो रहा है. A specific amount of blood in our body is getting purified almost 300 times passing by the nephron. Okay. So we are understanding ultrafiltration. What do we write about it? What is ultrafiltration? Ultrafiltration is the filtration of the blood happening in the glomerulus. Why the word ultrafiltration? क्योंकि ये जो filtration of the water and the salts and the proteins सब जो निकल के आया blood में से and is getting collected in the Bowman's capsule and trying seeping down, the blood is actually giving away so much of water. Almost about 90% of the water is given away during the process of ultrafiltration. So much of the protein, so much of the salts given away by the process of ultrafiltration. And then we get the glomerular filtrate. This is the first step of urine formation. But एक major question ये है, यहाँ पे you all should have asked me one question. How we are dosing. I'm constantly talking about this as ultrafiltration, a major step of urine formation, and etc., etc. And this is the composition I write. Did I ever mention about urea? I did not mention about urea at all, because by the process of ultrafiltration, urea can never come out of the capillaries. Urea can never come out of the capillaries because of the size of the urea molecule, जो pore size से much bigger है. During the process of ultrafiltration, what is happening? All the water, the molecules which can fit the pore size can definitely come out. So proteins निकल के आ रहा है, काफी सारे salts and molecules निकल के आ रहे हैं, काफी सारे proteins, some few proteins also cannot come, but yes, large amount of proteins, salts, sodium, potassium, all this can easily come out. RBCs definitely they get stuck. But big molecules, if I talk about complex molecules like the urea, uric acid, they also get stuck. They also cannot come out. They also cannot come out. So now, the blood which is moving away this side, should this blood have a high concentration of urea?
Yes, ma'am.
हाँ कि ना?
Yes, ma'am.
Yes.
What I told you, यहाँ से जो blood आ रहा है, try to understand. Otherwise, मेरा तो story खत्म हो जाएगा. You all will be blank. Try to understand. Don't think anything. Quite a number of you are distracted. I see every time. Try to understand what I'm speaking. This is the renal artery, the afferent arteriole I told you, which is carrying oxygenated blood. But the blood is rich in urea, uric acid. This is the blood which is running at a high pressure. अब जब glomerular capillary से high pressure में run हो रहा है, what is getting leaked out? Bowman's capsule में क्या collect हो रहा है? Huge amount of water. Huge amount of water, salts, ions, proteins, sodium, glucose, all these substances are getting collected. But what is not getting collected is urea निकल के नहीं आ रहा, जो आना चाहिए. Urea निकल के नहीं आ रहा है यहाँ पे because urea, the molecules are complex, they cannot match the pore size. RBCs also नहीं आ रहा है, but we are not talking about RBCs अभी हमारा यहाँ पे it's not important. So here we are trying to understand, does ultrafiltration give away a large amount of urea? No. Why no? Because the molecules are complex and cannot match the pore size of the capillaries. Then, अगर urea निकल के नहीं आ रहा है, can we consider ultrafiltration as a major step of urine formation? नहीं, इसमें तो अभी urea, uric acid निकलना बाकी है. So that's the reason, though this is a very, very important process of urine formation, this cannot be considered as the ultimate process. You get questions on this. Ultrafiltration is a major process taking place in the nephron. Can this be most important process for urine formation? It is most important, but it cannot be considered as an ultimate process because urea is not filtered out. Okay. So by the process of ultrafiltration, the blood which was coming by the afferent arteriole, as it passes through the glomerular capillaries, loses almost 90% water. 90% water, salts, ions is lost. Is the blood thick now? Is the blood which is running away in this vessel thick now? The blood which is coming this way, passing through the capillaries, has lost so much of water. And this is now known as the glomerular filtrate. So, ये जो blood अभी passing by the efferent arteriole, is this blood thick now? Is this blood thick now?
Yes, ma'am. क्योंकि सब पानी मानी सब
90% water it has given away. Is this not blood thick? It is thick. Blood thick मतलब क्या? It has given away so much of its water. Is this blood having high concentration of urea now?
Yes.
हाँ, urea निकला यहाँ पे? No. Ultrafiltration never gives away, never filters any urea. So since urea निकल के नहीं आ रहा है, the blood which is going to the efferent arteriole still has a high concentration of urea. So right now here you will have to compare. The first thing that you have to compare, in the efferent arteriole, the blood is thick. The blood has less water. Blood is thick when I say, what is the meaning? Blood is thick, it has less water and high urea. Clear है? The blood which was coming by this way had so much of water, urea, salts, proteins. But जैसे ही blood has passed through the glomerular capillaries, कौन से components blood का filter हो गया? Huge amount of water has come out, glucose has come out, salts has come out, proteins has come out. Urea did not come out. So now the blood which is going into the efferent arteriole is thick. Now the blood which is going into the efferent arteriole has huge concentration of urea. Now let us continue further. So ये हो गया process of ultrafiltration. Try understanding. I'll give you few questions. This is what is ultrafiltration. Where does it happen? What does it give rise to? What is the difference in the blood of the efferent and the efferent arteriole? It should be very clear. Now I talk about ये जो component which is running down, which is this component known as glomerular filtrate. अब ये glomerular filtrate is running down in the entire tubule. For understanding, let me differentiate the three parts of the tubule. Let me talk about the three parts of the tubule something very clearly. And here I say that this plasma, what does this glomerular filtrate contain? Plasma with proteins, salts, and huge amount of water. Always to remember this, large amount of water. What is this filtrate known as? This is known as the glomerular filtrate. This entire tubule, let me just divide into three parts. What are those three parts? Proximal convoluted tubule. I just write in short because we have to understand these two processes. I talk about the loop of Hanle and I talk about the distal convoluted tubule. Tubule के three parts क्या है? 1, 2 and 3. So now that the glomerular filtrate is running down, which is this fluid I am thinking of? This fluid is the glomerular filtrate. Glomerular filtrate is the filtrate formed after glomerular filtration, which is running down the tubule. Now I am to compare ये जो capillary में blood है. See, we have drawn the diagram this way. The efferent arteriole divides into a network of capillaries. So whatever blood is flowing in this capillaries, again वही दो parameters याद रखना है सबसे. What are the two conditions of the blood here? The blood is thick. The blood has more urea. So I am talking about at this feature, the feature of this blood vessel. The feature of this blood vessel is what? The feature of the efferent arteriole is blood is thick. Blood has more urea. Okay. So now that these efferent arteriole divides into a number of capillaries, now I'll have to understand when these blood vessels are surrounding the PCT, कौन सा process हो रहा है? Now that I'm talking about the blood capillaries surrounding the loop of Hanle, कौन सा process हो रहा है? And now that I'm talking about the blood vessels surrounding the DCT, कौन सा process हो रहा है? That is what next we are going to understand. ये दो process, okay? Selectively absorption and tubular secretion. So now I draw this blood capillary for my understanding. So let us talk about this blood capillaries जो tubule के आसपास है हमेशा. Which are these blood capillaries I am drawing? All these secondary capillaries I have drawn. So now talking about these secondary capillaries, the composition of the blood. Try understanding this part. एक बार समझ में आ गया, you will be able to write on your own. So now that the blood is here, the blood has got less water. The blood has got high urea. ये दो features याद रखना है. The blood has less water, the blood has more urea. Now let us talk about this. The first is I want to understand what is the process happening when the fluid is here. Fluid मतलब कौन सा? Glomerular filtrate. When the fluid is here and the blood is here, meaning I am talking about यहाँ पे क्या हो रहा है? All throughout the PCT and blood capillaries के बीच क्या हो रहा है? PCT and blood capillaries के बीच, the PCT has so much of water. PCT में बहुत सारा वाटर है, ब्लड में वाटर कम है. PCT में बहुत सारा ग्लूकोज है, PCT में बहुत सारा सॉल्ट्स एंड आयंस है, जो ब्लड में बहुत कम है. I already mentioned blood has less of water, high of urea. So let me compare. I would say when I try to understand a comparison, PCT और उसके आसपास blood capillaries में क्या कुछ processes हो रहा है? Yes, it's a major process happening because blood has got more of water here. I'm sorry, filtrate has more of water here. Blood has less of water here. So what is the process? The water will always diffuse towards the blood. The membranes, and these membranes are thin to allow a process of diffusion. Now the rule for diffusion you had come across earlier also, movement of a molecule from a region of high to a region of low. So ये जो water movement है from the PCT to the nearby blood capillary is a major movement. It's a major movement. So I already mentioned that यहाँ पे almost 90% मतलब ये जो fluid है ये बहुत ही dilute है. It's water, full of water, so much of salts and ions, and this is a thick fluid running here. अब हमारे बॉडी में अगर ऐसा thick fluid ज्यादा देर तक रहेगा, is that good? No. In few minutes, that thick fluid should again get back adequate amount of water. Otherwise, our body will start showing some improper functions. I'll continue again after the break. Don't erase the figures, let it be as it is.
तू आगे कुछ